Hello everyone. So for this week's lab, we're going to be trying to combine two experiments into one. So because of this, instead of having a lab report and a quiz due this week, we'll be having, a lab report and a quiz due 2 weeks from now. So because this lab report and quiz will cover basically two different chromatography techniques as well as SDS-PAGE, it should be very thorough. So I'm just letting you know now, please try to make these lab reports very lengthy because they should have 2 experiments worth of information. As well as when you study for your quiz, just know that the topics will be both chromatographies as well as SDS-PAGE. So this might make the quiz slightly longer. So to begin, what exactly is liquid chromatography? It's a very useful technique that we use that can purify proteins as well as other molecules based on some kind of specific attribute, whether we're targeting their charge, their size, whether they're hydrophobic or hydrophilic, or if we've manipulated the protein to have some kind of tag so that we can target the tag specifically. So this can be very useful because this allows us to really separate our proteins or our molecules or whatever we're testing on different parameters, and we can even use multiple chromatographies to really purify our protein to as clean as possible. So the way liquid chromatography works is you need to know that there's 2 different phases, a liquid mobile phase as well as a solid stationary phase. So we apply our sample into the liquid mobile phase, and this mobile phase will transport the molecules and transport the proteins, kind of dissolving them, making sure they stay in solution, until eventually they hit the solid stationary phase. Once they hit the stationary phase, this is where we actually get the separation. These solid stationary phase usually have some kind of beads that will basically separate our protein in some way. So the first one I want to talk about is ion exchange chromatography. So this is this week's lab. This is our actual first week's lab. And the way ion exchange chromatography works is it'll target the proteins and separate them based on their charge. When we talk about ion exchange, we usually talk about 2. There's either an anion exchange, which uses positively charged beads so that the negatively charged proteins will bind, as well as a cation exchange, which uses the negatively charged beads to target the positively charged proteins. So this means for our lab, we will be focusing on an anion exchange column, primarily because beta-gal is negatively charged. So why is ion exchange so useful? Well, if we have a sample of amino acids or a sample of proteins, and we apply and it goes through the column, what will end up happening is our negatively charged proteins will bind to the column, and the positively charged proteins will just flow right on through. So that's great. So we can separate our negatively charged proteins from both the positive and the neutral proteins. Something else we can do is we can actually separate some of the negatively charged proteins from other negatively charged proteins based on how negatively charged they are. So we do this by, using a wide salt gradient. So remember, salt, NaCl, contains a positively charged cation and a negatively charged anion. So positively charged sodium, negatively charged calcium, chloride. So if we apply sodium chloride into this column, the negatively charged chloride anions will start to take the place of these negatively charged proteins. So the more sodium chloride we add, the more chloride anions we add, we slowly start to push off some of these proteins until eventually we push off everything and the whole column is just negatively charged chloride anions. So this means as we apply a salt gradient at lower salts, we should expect the weakly charged proteins to come off first before the more strongly charged proteins. Also, in a perfect world, we can kind of manipulate the protein's charge. And we do this by really understanding the isoelectric point and choosing a correct pH for our buffers. So remember, amino acids can come into 3 different forms. a cationic form where the amino group has an extra hydrogen, giving it a positive charge, an anionic form where our carboxy group loses a hydrogen and becomes negatively charged, or is a Zerwitter ion where the charges end up canceling out and so it's basically neutral. So depending on the buffer that we use, or specifically depending on the pH that we put our proteins in, we can kind of control which state our protein will be in. So imagine if we are using a very basic solution, a pH of 8, 9, 10, we can expect that the buffer will contain lots of hydroxide anions. And because of this, this will pull off any free hydrogens from the amino group, causing our Zerwitter ion to go towards the anionic state, so it's negatively charged. Vice versa, if we use an acidic buffer, so a pH of 3, 4, 5, 6, we can expect the buffer to have many free hydrogens, which will bind to the carboxy terminal, or carboxy end of the Zerwitter ion, and give it a, remove the negatively charged so that it becomes more positively charged. So this is how we're going to kind of manipulate the charge of our protein. We know that if we work with proteins though, we need to keep the pH as close to physiological as possible. So that means 7.5. So if we use a buffer at 7.5, luckily beta-gal will actually be in its anionic state. So that means that our buffer is basic enough to form an anionic charged protein. So once we separate off of an ion exchange column, we actually look at it under a gel, but we'll talk a little bit more at the gel later. It's because the next kind of chromatography I want to talk about is size exclusion. And this one's a little bit simpler. It's kind of like how we worked with the agarose gels, where our proteins will move through the column based on their size. The only difference is that larger proteins will actually elute first, and the smaller proteins will elute last. So this diagram doesn't really depict it very well, but you can kind of see these little openings in the beads. So what will end up happening is our big proteins will just kind of bounce around and go around all of these little beads, which is how they kind of get through. While the smaller beads will kind of get trapped, so they'll go in here, get stuck a little bit, and this will slow them down. So this is how we kind of separate all of the proteins based on their size. So this one's very simple. Like I said, we're going to be using both ion exchange as well as size exclusion to purify our beta-gal to make sure it's as clean as possible. So now this leads me to the last part of the lab, which is the SDS-PAGE, or the sodium dodecyl sulfate polyacrylamide gel electrophoresis. So this works almost exactly the same as our DNA agarose gel, where we're going to pull a sample down to the bottom, and as the sample runs through the gel, it'll separate based on its size. So just like the agarose, we can expect that the smaller sized proteins will move down the gel faster, while the larger sized proteins will move slightly slower. So here you can kind of see that there's two different layers to the gel. The first is a stacking layer, and the second is a resolving layer. So the purpose of the stacking layer is so that when we apply our samples into these wells, they kind of move through the stacking well together and will actually, the whole well, so this giant well right here, will kind of condense itself until it becomes a very thin line. And then the moment these thin lines reach the resolving gel, that's when we can get everything to separate. So the resolving gel is the actual gel that will separate our proteins based on their size. So you can see here that we have two different pHs for our gels, and this is to really manipulate that whole condensing of our protein sample. So outside you see that in our buffer we're using tris and glycine. So glycine is kind of the important factor here. The reason being is that when glycine is put in a pH of 6.8, it becomes a Zwitter ion. So it becomes neutral. So you can see that there is an anode and a cathode. So things that are negatively charged will kind of pull themselves down through the gel. So when glycine is neutral, that slows it down. So because we have a ton of buffer in the sample, as our sample runs down, it wants to kind of go down as quickly as possible, but the glycine is holding it back. It's kind of pushing back upwards. So we have the glycine pushing upwards, we have our protein sample pushing downwards, and this is what's condensing our sample into that thin band. So because the protein sample is a bit stronger, because it does have that negatively charged, it isn't. So remember, glycine isn't quite repelled, it's just moving down slower. So we can expect that our sample, as it goes down, the band becomes thinner and thinner and thinner and thinner and thinner until eventually it reaches the resolving gel as a line, just a very thin line. And then once all of our samples are this thin line, they run through the resolving gel at the same time and can be separated the same way. This way we can kind of actually look at compare our samples to a ladder and really understand that whatever we're looking at is very accurate. So one, issue is, what if we have proteins that are positively charged? If we just apply a sample that has positively charged proteins, then they'll go up. They won't go down the gel. So we have to treat our sample not only to give them a negative charge, but to also linearize them. We don't want big clumpy complexes to go through the gel as a complex, because they just won't go through. But if they're linearized, then they'll have a much easier time to go through the matrix, and we'll actually be able to see them on the gel. So to treat our proteins, we do three basic things. First, we heat the proteins. Adding heat will break any kind of hydrogen bonds, which will weaken the protein structure. We also add a reducing agent. For the lab, it's going to be beta-mercaptoethanol, and this will break down any disulfide bonds. Finally, where the SDS comes in. So in the buffer is sodium dodecyl sulfate, and it'll coat the protein. As you can see, it'll coat the entire protein, and it'll give the protein an entirely negative charge. This way, we can run not only negatively charged proteins, but also neutral and positively charged proteins on an SDS-PAGE gel. So that's pretty much the end for the theory for the two labs, or the two techniques as well as SDS. I'm going to try to have everything on. So I'm going to put the lab material this week, but I'm going to hold off on putting the quiz and the lab report until next week. So remember, the lab report and the quiz are due 2 weeks from now. And with that, I hope you guys have a good day.